So thanks, Alistair. Like Jane, I'm going to pitch my talk mainly at the parents and grandparents and carers in the audience. But actually, to be honest, often I think taking these things and asking these questions at the kind of starting from scratch level gets you more answers than focusing greatly on the technical details of how we do genetic testing or the sorts of pitfalls there are. Often the pitfalls are quite basic and general in terms of the approaches we need to take to try and find an answer and try and find an answer faster. This is a picture of the British Library room at the British Museum, that central round building within the canopy of the British Museum. And throughout the talk, I'm going to be using the analogy of a library in terms of our genetic information and the way it's stored and the way in which we need to search for an answer to the cause of what we consider likely to be a genetic condition in a child or adult with complex medical disorder. And as Jane has said, what we're good at is finding an answer where there is one genetic cause. We don't always find it in that scenario, but that's the scenario where we're strongest at finding an answer. But that isn't always the case in children with complex conditions. As Jane said, there can be environmental causes where, as hard as you look through the genetic code, there's never going to be a simple answer that you find. It might be that there is a genetic component to it and there's multifactorial cause. So, for example, Jane talked about cleft lip and palate can have that scenario. And while you'll read research papers where they say, oh, this variation makes cleft lip and palate a bit more common or similar with spina bifida, there won't be a simple answer that tracks with the question, will this child be born with this problem or not? It might gently influence the likelihood in the family. And those sorts of genetic variations might be something that could be looked for in a research study and published on the level of a population, but aren't going to be useful, certainly at the moment, for us and for families in clinic wanting an answer, what's the cause, why has this happened? So if we start off with our analogy of this library, our genetic code, you'll know, is written in a language called DNA. And it's a language which, rather than 26 letters and a bunch of punctuation, just has 4 letters, A, T, G, and C. But it's still a language, just like our written language. The way the information is written is, if you like, in sentences, and it tells the body how to do all of the different things that it needs to do to grow and form and develop and maintain itself. The way the messages are written are a little bit like in books. So genes are the equivalent of books in a library. Each gene is written in thousands of words and divided up into chapters, if you like, which are called exons. And that gene will have a particular message. It might tell the body how to produce a particular building block for the body, a protein, a particular building block that does a very particular job, perhaps to do with the transmission of messages between nerve cells in the brain, so that when there's a problem with that one particular book, there's that particular problem with the body. Perhaps if that was one of these genes that is involved in messages being transmitted between cells in the brain, it might predispose to epilepsy if the gene had an error in it. Now, just like a library, we have lots of genes. In fact, we've got about 20,000 different genes in the human genome. And as Jane said, we've got 2 copies of each of those genes, one inherited from each of our 2 parents. It's like having a first copy and a backup copy, if you like. They're arranged on these structures called chromosomes that Jane showed you a picture of, and you'll see in a little bit when I'm talking to you about how we do testing. Chromosomes are a little bit like bookshelves, so that the genes, the books, are individually packaged one after the other on these chromosomes. And some people in clinic, particularly when we found a chromosome abnormality on chromosome 3, will ask the question, well, what does chromosome 3 do? What's that important for? Well, these books haven't had a careful librarian sit down with them at the beginning and think, well, we're going to put crime over here, we're going to put personal development over here. They've just been all dropped on the floor and then put back on the shelf in a hurry, so that a gene that is important for brain development might be right next to a gene that's important in thumb development. Sometimes we can know when we look in a very detailed patch of a chromosome, we'll say, well, that's one that's particularly involved in this sort of area of development. But actually, genes are very randomly arranged generally. But it is useful for us to know where they're stored in the human genome, in this library, because sometimes it helps us find them, and to find where the fault is, where that genetic alteration is that is causing the problems, if there is just one genetic alteration causing the problems that we're seeing in the child. So this is a picture of a chromosome result, and Jane's already shown you these chromosomes, which, as I said, are a bit like the bookshelves in the library. They come in pairs, and there are 46 of them in total, and there are 23 pairs. And when we're thinking about these sorts of disorders where I've told you we've got a relatively higher chance of finding a definite answer to the question, well, why has all of this happened? There are different levels of sort of genetic problem we might find. We might find a problem at the level of a whole chromosome. So, for example, a whole extra copy of one of the chromosomes, as is the cause of Down syndrome, a whole extra copy of chromosome 21 sitting down here. But at the other end of the scale, we might be looking for a problem at the level of a single gene, and not even a problem with that whole gene, that whole book. It might be a problem with a single letter within that book. And if you think about the scale of the problem of identifying that single letter change in a book within this large library, that's the challenge we have. And we have to, at the moment, target a lot of our testing to tell ourselves, well, we can see this group of problems, this pattern of problems. Well, I think it's probably going to be this book, or amongst this group of books that we know can cause this sort of pattern of problems. But we've been lucky for some years that we've had some less targeted testing that has given us answers without us having to be so targeted, so clever, if you like, in terms of jumping in and saying, well, I think this is likely to be the answer. And that's chromosome testing. So the equivalent of chromosome testing, I guess, is standing in the doorway to the library and saying, well, let's just take a bit of a look around. Can I, without jumping to any conclusions, can I spot where the problem, where the genetic alteration that's the cause of this condition lies? And that's what we call a non-targeted or a genome-wide test. And chromosome testing is a type of genome-wide testing. The problem with it is when you stand at the door of the library, you can only make out the shelves in quite general shape. And you might be able to see that there's one missing, or there's an extra version of one of the shelves from that distance. So you can see that maybe you can see something on chromosome 11. If you're lucky, you might be able to see that there's a little missing chunk within the chromosome, rather than there being a whole problem with the whole of that chromosome. And that's been fantastic, because you don't have to be so specific in targeting the testing. If you just say, well, let's do a chromosome test, in quite a lot of those chromosomal disorders, for years we've been able to jump to the answer. Chromosome testing itself, though I'm saying, we can quickly jump to an answer, still takes a couple of weeks at least to get an answer, and normally more like 2 or 3 months. And the reason for that is that you have to get the cells that you take from the person you're testing to grow, so you can see the chromosomes as the cells divide. Now, chromosome testing has got a lot better in the last 5 or 10 years with the introduction of this technique called microarray testing that we've already heard about. And so that rather than using a microscope to look down at these chromosomes from afar and literally look with your own eyes, or with the chromosome expert's eyes, to see if there's an extra copy of that chromosome or if there's a missing chunk or an extra chunk that you can actually see, this microarray testing has a much finer level of detail that it gives. So it will spot the really obvious changes that a microscope could pick up, if you like, from the doorway of the library. But actually it can zoom into a far higher level of detail, and perhaps it can pick up a sort of detail where just one of the shelves is missing or with an extra copy. So there's a missing or extra copy of a few genes there. And that's really revolutionized our ability to pick up smaller chromosome alterations. And the level of detail it gives is perhaps 50 or so times the level of detail we used to get earlier on with chromosome testing. And so, as Jane said, when that testing came in, suddenly there was an additional group of families where before we'd always been saying, well, I think there must be one cause. And sometimes we'd even said, I think it's probably a subtle chromosome change, but we just hadn't had the ability to do that testing. Suddenly, when this microarray testing became available, initially through research, and now very routinely in NHS genetics laboratories, we can just jump to finding those abnormalities. And because, like chromosome testing, it's what we call a genome-wide test, it doesn't require us to say, oh, you know, I think this is going to be on chromosome 7. It will give us the answer. So that's chromosome testing and how it's changed. Single gene testing is where we're looking down right at the finest level of detail. We're looking down at the level of a single book in the library. And actually, as I said before, often the problem when there's a single gene that's causing the condition isn't at the level of the whole book. It's not that that whole book is missing, although that can be the case. It's not even necessarily that there's a problem with a whole chapter or a whole page. It might be that there's a single letter that's altered in that book that's changing the spelling of a key word so that when the body's machinery reads the book, it can't make sense of the words and therefore can't use the book to do the job that it's supposed to be telling the body to do. Now, I'll come on in a little minute to talk to you about the newest genetic technologies, which are the sorts of technology that you'll hear a bit more about from Maria and Howell, and that are being used in the DDD study to look in a genome-wide way at this single letter code level of detail of the genetic message. But up to now, and still for a lot of the genetic testing that we do in the NHS laboratories and a lot of the testing that's done on a research basis, we have to target our testing to one particular gene at a time to ask the question, is this one gene the cause of the problems in this particular person? And the way that that testing is done is to literally take that book and read it letter by letter. And that's been done by a technique called DNA sequencing, and has been the same technique that we use to read the whole of the human genome as part of the Human Genome Project. And has only, that very first type of DNA sequencing has only just been replaced by these next generation technologies. It's a fantastic technique, but it is laborious. The problem with it is that rather than even just pulling out the whole book and reading that all at once, sitting down for an afternoon and getting through and really having a close eye and thinking, well, you know, I've got to be paying really close attention here because it might just literally be a single letter that's changed or a full stop that comes too early. You can't read the whole book in one go, in one test tube with the technology that as it was. what you do is you read the book in little chunks, and those chunks are only about 300 or 400 letters long that you can read in one go, in one little test tube. So when you see on the television someone with one of these little trays with lots of little wells in it, like lots of little holes in an egg, or lots of little egg cups. Each of those one little wells is like a little test tube, and each of those will read for you those 300 or 400 letters. So you need a little well for each of those chunks of the gene. And the gene might be 2,000 or 3,000, maybe 10,000 letters long. So you can imagine that costs more for each of those little reads that you get, and it just takes longer as well. And it's that sort of testing that we've relied on and still rely on for a lot of the genetic tests or single gene tests that we do. Because the testing is expensive and quite laborious, what often happens is that testing for one gene might take, again, 2 or 3 months for all of those tests to be done. The scientists in the laboratory to sit down and read through, double check that they're reading it, that they've not skipped a letter that was the answer. And also to go back, and when they find a subtle spelling change in the book that they're not quite sure when they first read it is important or isn't important, they perhaps go through all of the databases that are now available of known spelling mistakes, if you like, which can cause the condition we're talking about, or known spelling variations that might be normal, because we all differ at an awful lot of places across our genetic code, and that's what makes us all different. Even within one ethnic population, we vary a lot, even within genes that can cause diseases. So even when we were just looking at one gene at a time, a lot of the scientists' time wasn't spent with that pipette like you see them on the telly. It was sitting in front of a computer, checking they'd read everything nice and accurately. But also when they'd found some spelling differences, they were consulting databases, perhaps going back to the samples from the parents and trying to answer the question, well, I found this spelling change here. Well, but if I read it quite fast, it kind of sounds a bit like worst still. Maybe it doesn't cause the body to misunderstand the sense of the sentence. Maybe this isn't the cause, just because there's a spelling change there. So that slows things down as well. And as Jane also told us, it's not always as simple as saying, well, we know this gene can cause this condition. For example, epilepsy. We know there are an awful lot of different genes that can cause conditions which predispose to seizures and are labelled as epilepsy disorders. So I'm sure a lot of families in the audience have been in the situation where a good number of single genes have been tested one after the other using this sort of technique to try and find an answer. And it may be that if we are fortunate, after 3 or 4, an answer was found. But because of the time it takes to read each gene, that's even when you do jump to an answer, it may be that some years have passed. What's changed in genetic testing in at that fine single letter code level in the last few years is absolutely amazing, and has really changed the way genetic laboratories and our ability to find answers where we haven't been able to before and find answers faster. It's really revolutionizing things. It hasn't yet got to the level where, for example, with microarrays, they're just something that we're very au fait with and they're routinely available in the NHS. But there will come a time in a few years where, rather than relying on this sort of new testing through research studies, it's something which is a routine NHS diagnostic type test. But you can probably, in the things I've told you about even the single gene testing, start to see what a difficult task it is to interpret genetic tests for one gene, let alone a whole library's worth of information. So these new genetic testing technologies that you'll often heard referred to as next-generation sequencing And really, it's only the second generation of sequencing. There might be third and further generations which really even further speed things up and help us. Instead of standing at the doorway of the library and just being able to say, oh, I can just about see that there's a whole shelf missing over there, or perhaps just within that shelf, just one line of books missing. This allows us to read right down to the single letter code across the whole of the library. Now, the problem there becomes completely different. It's not the ability to get the information across all of those genes and to target the testing. This is a genome-wide, or at least when we use the term exome testing, what we're doing is just testing the genes, the coding part of the genome, and I'll come back to that in a tick. The problem isn't just getting the information on whether there are spelling differences in the person you're testing compared to what we know is common in the general population. The problem is information overload. Suddenly, instead of you finding it difficult to get the information onto your computer as a scientist by having to laboriously stand there with all of those test tubes, you can do this, it's not quite a test tube, but effectively in a single test tube. The problem is then the other part of the equation, which is how long you have to sift through all of that information on your computer, how you need to be very careful that you don't overinterpret variations that you find in, as I've told you, even the genes that we know very well and have known for years can cause certain diseases. It's hard enough when you're looking at one particular gene and trying to interpret things. You have to be quite cautious. When you've got all of that information flooding to you on 20,000 genes, the problem is that interpretation stage. And that's what the DDD study and other exome and subsequently genome research studies really are teaching us more than the technical side. It's how we process all of that information. The last thing that we want to do is be over eager in thinking, oh, yes, we've seen something which probably is the cause. Let's just say that's the cause and move on and not think more carefully. It's very important that we're cautious in interpreting the information that we get and give results that truly are the answer to what's the cause in this child. Because actually, an awful lot of harm can be done by getting it wrong. For example, advising people that there's a very high risk of a condition happening again because we think that we've found evidence that there's perhaps a certain pattern in inheritance going on as the cause, where in reality the cause isn't the thing that we've been looking at. It's something else entirely that we've dismissed as an innocent variation, which has started for the first time in that child and has a very low chance of happening again. So from the DDD study, for example, and other exome studies, we know that this is a good way of jumping to finding the answer in single gene conditions that previously we weren't able to target our testing to before. And as an aside, while these exome, looking across all of the genes, while these exome studies are going on, the other thing which a lot of NHS laboratories are also doing in parallel is, if you like, a halfway house to that, which is we refer to as gene panel testing. So rather than the more difficult to manage task of looking across all of the genes in the library, we're selecting groups of genes, for example, all of the the testable genes that can cause deafness, and looking for variations in those genes in a child where hearing loss is a problem, or the genes that we know can cause early onset seizures, and just looking at those. So that's speeding up our testing. It has some of the problems that you encounter when you look at an awful lot of genetic information, and the interpretation stage, it slows things down, but it is taking things a little bit faster. You'll also have heard, though, about whole genome sequencing, and that sounds like, well, surely that's going to be the answer. That looks across all of the genetic information. Now, well, we've been talking an awful lot about genes at the moment, and it often surprises people to hear that amongst all of the genetic information in the library, only about 1 or 1.5% of that information is actually in the genes, in the books that we understand, tell the body how to produce these individual building blocks that make up the body and maintain it. The rest of the information has previously been dismissed as what we call junk DNA years ago. And it was dismissed as that really because we didn't understand, and we still don't understand, how most of it works. But we know that there must be elements of it, and probably quite large sections of it, that are important, that do have functions. But what we struggle with at the moment is interpreting how those bits work, and therefore how any variation in those other bits of the genome work. So it might be that there's an answer hiding elsewhere in the library, not within one of those genes. And that's why genome sequencing might be better at finding an answer than exome sequencing. But our problem there is even more amplified in interpreting what's an innocent variation that is there in a difference between any two of us, and what is the important cause. So the final thing, which being us gaining further experience with using these techniques, and the more people who take part in these research studies will give us, is that one thing that we're better at jumping to is an answer where the answer lies in a gene that we've got a lot of experience in before. There are an awful lot of conditions out there we still know, which we don't know the cause for, and where when we meet the first child and perhaps even genome sequence their DNA with that condition, even if we find that answer, we won't be confident it's the answer until we meet further children with the same condition who share a change in that same part of the genome. And that's the other element, the 21 years between first seeing the patient in clinic and the inbox having the paper going out for publication where you can be confident you found the cause. That can be the longest wait, is to find other people with the same condition that give you the confidence that you found that answer. So I'm going to finish there.